Very good evening, everyone. Please confirm if the screen is visible. Am I audible? Can you all see the screen? Good evening, Rahul. Good evening, Hina. Very good evening, Anupam, Aditya. Okay, thank you all for confirming. I can see Samura joining. Good evening, Kirtana. So, welcome to another session of Brahma series, the session on Unit 8 questions. So already we had the we already have a session where we discuss the questions from Unit 8A, that is Mendelian principles, probability questions we discussed, Mendelian ratio based questions we discussed. And today in this particular session, I will be discussing the questions from subunit 8B, that is concept of gene, and 8C, that is extensions of Mendelian principles. So I'm sure you very well know the syllabus of Unit 8. I'm sure you know that 8B, when we talk about 8B, it comprises of terms like multiple allele, pseudo allele, complementation test, right? And specifically from exam point of view, the questions are asked on multiple allele, specifically the blood group. And also the questions are asked on the complementation, right? Complementation test. So such type of questions I will be discussing today based on these topics. And also questions on extensions of Mendelian principles. So extensions of Mendelian principles, this particular subunit, it includes many terms like incomplete dominance, co-dominance, and you have epistasis, pleiotropy, phenocopy, right? Many terms are there. And it is very much important for you to be familiar with each and every term, because you never know in the exam the question is asked can be asked on any of the terms. Even genomic imprinting is also there. So today I will be discussing the questions that has been asked in previous year question paper from these the topics from these particular subunits, and also I will be giving you certain questions, the type of questions which can be expected in the coming years of the coming CSR net exam. So let's get started. Let's start with the very first question. I'm sure you all are ready. I can see some more have joined. Very good evening, everyone. So I'm sure you all are excited. Yes, excited to solve the questions, excited to discuss the questions. Yes, please mention yes or no in the chat. Okay, that's great. So the very first question you can see on your screen is, it is about a newborn baby of a mother having blood group AB Rh positive and father having blood group O Rh negative got mixed with other babies in the hospital. The baby with which of the following blood groups is expected to be of the said couple? So four options are here. And I'm sure by looking at the question, it is very much clear. The question is about the blood group, right? And we know that there are multiple alleles coding for the blood group, many alleles coding for the blood group. So you have to recall that concept here and you have to solve the question. So let me see. You all can try and let me know what is the answer here. Okay, some of you have already solved. So Anupam and Rahul, according to you, it is option 4. Fine. So let's others also solve and then I'll discuss the answer. I'll reveal the answer. Okay, Suresh 4, Aditya 3, Kirtana 4. Okay, so many of you are saying option 4. Let's see what others have to answer. So here you have to consider the alleles for the blood group as well as you have to consider the alleles for the rhesus factor, RH. Okay, Anupam, you're asking that why we don't get such easy question during exam. Anupam, this is the question from previous year question paper only. This is the question, part B question. Okay. This is the question I have not framed myself. This is the particular question I have taken from previous year question paper only. I am not this is not from the book. Okay. So you get such easy questions. The point is you get such easy questions. Okay. So Tulsi, Anita, okay. Ankita, you are saying option 4. That's great. So let's check. So first of all, as I told you that you have to recall about the concept of blood group here. So I'm sure now you all know because I have already taken this topic in the class. So I'm sure you know that there are three common alleles for ABA blood group. So we have one allele which is coding for A antigen, another allele coding for B antigen, and then there is an allele which does not code for any antigen, right? So basically, if the individual gets the allele coding for A antigen, right, both the alleles from the parents coding for A antigen, blood group is A. And if the individual gets allele coding for A antigen from one parent, but allele coding for no antigen from another parent, then also blood group is A. Why? Because it is observed that the allele coding for A antigen is dominant over the allele without any coding for no antigen. Similarly, when you talk about B blood group, so B blood group is possible if the individual inherits the allele coding for B antigen from both the parents, or one allele coding for B antigen from one parent and allele coding for no antigen from another parent. So why is it so? Because we know that allele coding for B antigen display dominance over the allele coding for no antigen. Now, O blood group is only possible when the individual inherits the allele coding for no antigen from both the parents, right? So because this particular allele is recessive, it's a recessive, like allele for A antigen and B antigen are dominant over allele coding for no antigen. And when these alleles, like one allele, if an individual inherit allele coding for A antigen from one parent and allele coding for B antigen from another parent, then in that case, the blood group will be AB because they show codominance. Now, since it is mentioned in the question that mother blood group is AB, right? So that means the mother must be having this genotype, right? And the father blood group is O. So as I showed you in the previous slide, that the O blood group is only possible when the individual has the recessive allele in homozygous condition, that is the allele coding for no antigen in homozygous condition. So basically, if you draw the, like if we see what combinations we can get in the progeny, so this will be the gametes of one parent, this is the gametes of another parent. So we'll get this combination in the progeny. So either we can get A blood group or we can get the A blood group. Either A or B blood group only is possible, as you can see here. Now, I told you we have to consider the genes for the blood group and also we have to consider the gene for the rhesus factor. So it is mentioned that mother is having the Rh plus, but father is Rh negative. Now, let's consider this D allele to be the dominant allele and coding for Rh plus factor, right? But the recessive allele, it is for Rh negative. So basically, as you know, that father will be contributing only this allele. But here, if the mother is homozygous dominant, so we know Rh positive is possible if mother is homozygous dominant or heterozygous. So if the mother is homozygous dominant, then we'll get this combination, heterozygous condition in the progeny. Again here, depending upon whether homozygous dominant condition we get or the recessive allele we get, accordingly, we'll get again 50% heterozygous condition or 50% homozygous recessive. So you can see there is a 50-50% chance, 50% chance for A blood group, 50% chance for B blood group. Similarly, if you talk about rhesus factor, so 50% chance for Rh positive and 50% chance for Rh negative. So because there is an equal chance to come, so either the A blood group can come, which can be Rh positive or Rh negative, or we can have a B blood group which can be Rh positive or which can be Rh negative. So as you can see the options, in the option only B Rh plus is given, Rh positive factor is given. So therefore answer here is option 4. So I'm glad most of you have answered it correctly. So now moving over to the another question. So let's have a look at this question. If two mutant strains of bacteria are crossed and all of the resulting progeny exhibit the mutant phenotype, what can be concluded? So here again, you just have to recall one particular concept. If you are able to do so, you can easily answer this question. Such type of questions, students, these are very easy and they are asked in part B, right? Like, even in part B also you can score. In genetics, usually in part B, we get 4 to 5 questions and part C, we get 4 to 5 questions, right? So just imagine, like maybe you're finding part C questions a little bit difficult. Let's say you are not able to uh, solve the probability question, right? Like you practiced, but you all know that in the exam questions, probability, even we talk about different types of questions can be asked from probability, right? So maybe in the exam you feel like that, okay, this question I cannot answer. I'm not sure about the answer, right? Like you're not sure about the answer. So in that case, try go for part B questions, right? In part B you can score well in genetics. So the point is when you're preparing the topics for the exam, emphasize on these topics from part B and part C. Don't skip this part B and part C subunit. This unit, these two subunits of unit 8 can help you score better in the exam, right? So I'm telling you what type of questions can be asked from these, from the topics of these two subunits and what you need to prepare, right? So here you need to prepare the concept of complementation. If you are able to recall that, you can easily answer this question. So let's have a look at the answer. So the question is about two mutant strain of bacteria crossed and all of the resulting progeny exhibit mutant phenotype. So what do you conclude? That mutations are allelic, they are not allelic, or they are allelic, they are dominant, or they are recessive. Okay, now for this, I'm getting mixed answers. Some are saying option one and some are saying option two. So that is what in part B also you will come across the two statements in which I'm sure the confusion will be there. So how to overcome this problem if you are reading the two statements and you are getting confused? So how to overcome this, right? So I'm glad at least out of four you are able to. I'm sure you are able to omit two options out of four, and I'm sure you all, for some it is option one, for some it is option two, right? So as I mentioned earlier, you have to recall the concept of complementation here. So Kirtana, Rahul, Kantimati, Suresh, Vanishri have already answered, and I can see many students are here, but they are not answering. Students, please tell me the answer. It's for your own benefit only, right? At the end, in the exam, you have to solve such type of questions, right? So I can only help you if you let me help, right? If you allow me to help. So you need to answer the question, then only I can tell you that whether whatever you are thinking, whatever concept you are thinking, and what answer you have marked, is it correct or not. Vanishri, you're absolutely right. I will be discussing about that, complementation and is it allelic or non-allelic, how to find. Okay, Tulsi, option one, Anupam one, Yusra, you are saying two, and Sojani, you are saying non-allelic. Fine. So as I mentioned few minutes back that here you have to recall the concept of complementation. So if you remember, complementation test is done to determine whether two mutations are in the same gene or different gene. So basically here you can see the two genes D and R, and maybe these genes, like these genes are affecting the same character, right? Like one example, I'm sure you all know Drosophila, right? The eye color, the eye pigment in Drosophila. So many genes are there which are affecting this particular trait, that is the eye color in Drosophila. So wild type is red, but we have the mutants also which are white, which are an apricot, right? So maybe these G and R are the two genes which are affecting the same character. Now you can see that in this first figure, you can see that M1 is the mutation and M2 is the mutation, but this mutation is in the different gene, right? So always remember, if the complementation has to occur, complementation will always occur when the mutation are in the different genes, right? So here you can see that this M1 and M2, they result in wild type phenotype. Why? Because every, like if you see here, this particular chromosome is providing the wild type allele, and here this chromosome is providing the functional gene, right? So this is the reason the wild type phenotype is there. But if you see here, when the mutation is in the same gene, you can see the defect M1 and M2 is it in the G gene only. Here it was one defect in G, another in R, right? But here it is in the same gene. So this results into mutant phenotype. So this point you have to always remember, that when we are talking about the complementation, so here both the strains were mutant, and when they are crossed, they produce mutant phenotype only. So that means complementation has not occurred, right? And why complementation has not occurred? Because they are allelic mutations. They are alleles of the same gene. Complementation occur when we have the alleles of the different genes come together and they result into a wild type phenotype. Okay, Vanishri, you're asking, ma'am, how do we know the question is based on complementation? So, Vanishri, like even if you look at this question, you very well know that it is about the phenotype. Is it a mutant phenotype or is it a wild type phenotype, right? And if you look at the options, the terms are mentioned allelic, non-allelic, right? So you know that these terms you have studied with respect to complementation, right? Dominant, you know, if it is about dominant recessive, so dominant expression will come, right? Recessive allele can only express, a recessive phenotype can only occur when recessive allele is in homozygous condition. So here they have given you a condition that two mutant strains are there and they are crossed and finally they exhibit mutant. So this kind of examples, these kind of like problems we have seen when we were studying complementation, right? So it all comes with like, you know, how do you know that it is about complementation? So it comes, you're able to recall the concept only when you read the different examples of complementation, when you read about complementation. So you then in that case you can you know, correlate. I hope this answers your question, Vanishri. And allelic means that we are talking about alleles of the same gene. Non-allelic means when we are talking about the alleles of the different gene. Okay, that is the point you all have to remember. So moving over to next question now. So in snapdragons, red flower color, it is incomplete dominant to white, and the heterozygotes are pink. A red flowered snapdragon is crossed with white flowered one, and you have to determine the flower color of the progeny of a cross of the F1 snapdragons to the white parent. So here the options are given, whether you get 3:1 ratio, 3 red, 1 white, or whether you'll get 1:2:1 ratio, 1 red, 2 pink, 1 white, or whether you'll get half red, half pink, or half white, half pink. So you have to answer the question based on the information given to you in the question. Okay, Ashwin, you are saying option 1. Anupam, you are saying option 2. Okay. Sajanya, option 2. See, always remember, I have told you in the previous class also, the very first session also, when we were discussing about questions of probability, I told you, you always have to answer the question with respect to what information is given to you in the question, right? I'm sure while reading this question, you would have able to recall that this question is based on incomplete dominance, right? Because I'm sure the Snapdragon is a very popular example, and we all have studied this example in detail in the class also, right? So based on what you studied and based on what information is given in the question, that is different thing. Okay, what you have studied, you have studied about phenotypic and genotypic ratio of the uh, in F2 generation in case of incomplete dominance. But here you have to understand, it is mentioned that red flower is crossed with white flower, right? And then whatever F1 generation you have got, that you have to cross with white parents. So what you will get? So just emphasize on this particular part. Okay, now I'm getting option 4, option, okay, Kirtan, you are saying option 3 also. So 4, it will be 4 or option 2 or option 1. Just think about it. Take your time. It's an easy question. This is red flower, right? And so having this genotype, and white flower having this genotype. When they are crossed, you get the progeny that is heterozygous, and you get pink color snapdragons, right? So red flower snapdragon having this genotype, because it is mentioned this is the allele, right? And this is the allele for the white, right? So it is crossed with white flower. Now determine the flower color of the progeny of a cross of F1 snapdragon. So this is F1 generation. You have to determine the cross between the F1 generation with white parent. That is this cross you have to perform. And based on that, you have to answer the question. Draw the Punnett square. How you are getting 3 red and 1 white? It cannot be 3 red and 1 white, because you know this ratio we don't get in incomplete dominance, right? This is the ratio we get in Mendelian principle when the dominance, concept of dominance, right? The alleles which are displaying the dominant, complete dominance. Yes, it will be half white, half pink. So you very well know. So this is the example of snapdragon. And what is incomplete dominance? Incomplete dominance is when the heterozygote, that is F1 generation, has a phenotype which is intermediate between the phenotype of two homozygotes. So here we have the red and the white snapdragon. So the genotype is already mentioned. You know, parent generation is pure breed, right? So if you perform this type of cross, F1 generation will be what? All pink. It is already mentioned in the question. And obviously they will have the genotype that is heterozygous condition, right? Now, if you perform selfing of F1 generation, right? So selfing of F1 generation is where you have one plant heterozygous, another heterozygous. And in that case, yes, you will get 1:2:1 ratio, phenotypic and genotypic both. One red, two pink, one white. Fine. But here it is not about selfing of F1 generation. It is a cross where you are performing the crossing between the pink and the white. So I will answer the question. So in the question we have this ratio, this alleles mentioned, right? This is what is mentioned, the allele, right? So I will answer it accordingly, right? So this is the gamete formed by one parent. This is the gametes of another parent. So what you will get? This is heterozygous. This is homozygous recessive. This is again heterozygous, and this is homozygous recessive. So what you will get? These half will be what? Pink, and these half will be what? White. So half pink and half white. It was such an easy question. Why you were saying option 1, 2, right? It is option 4. Yes, absolutely right, Surendra, Tulsi, Saujanya, Anupam. Okay, Anupam, you're saying read the question. You have read the question half it was. So that is what, Anupam. Very good. I really appreciate you. You tried the question, and what mistake you identified, where you are making, where you are committing the mistake, that you didn't read the question completely. So what we learned today, that we have to read the question completely. We have to read all the information what is given. We have to read it carefully, and accordingly we have to answer the question. So I hope you will never commit this mistake again, right? This is the importance of solving the questions. You learn, right? You know where you are going wrong. Okay, moving over to another question. There is a researcher exposed Drosophila larva to 37 degrees during their growth. Now, one of the adult flies that emerged had a cross veinless phenotype. Now, cross veinless is a known mutant in Drosophila. See, this is all the information is given. When this cross veinless fly was crossed to non-cross veinless mutant fly, all progeny had normal phenotype. So what do you think? The observed phenotype can be explained as an example of, is it a conditional mutant? Is it a phenocopy? Is it a penetrance? Or is it pleiotropic? This question you can easily answer if you have studied this example and all these terms if you have studied. So answer this question quickly. Okay, Achal, you are saying option 4. What about others? Rahul, option 2 you are saying. Okay. Phenocopy, Surendra, you are saying. That is option 2. Okay. Yes, quickly answer the question. Okay, majority says option 2. Good. Okay, Suresh, you are saying option 1. So you have to like you have to know about all these 4 terms, then only you can answer this question, right? So here you can see that phenocopy, penetrance, pleiotropy, all these terms you study in subunit 8c, extensions of Mendelian principle, right? So if you don't study all these terms, you can never answer such questions, right? And such questions are very scoring. So it is very much important that when you are studying subunit 8c, extensions of Mendelian principle, you study all the terms, don't skip any of the terms. Fine. So majority says option 2, but even some are saying option 1 and 4. And I can see some are not responding. What happened? You are not able to solve. Please let me know if you're not able to solve. Just try first, okay? So what is conditional mutant? What is phenocopy? What is penetrance? What is pleiotropy? So let's have a look. Penetrance is what? Penetrance, it is measured in terms of percentage, where we say that whichever individual is having a particular genotype, it shows it. It is capable of showing it, right? Like it is the percentage of individuals that show a particular phenotype among those capable of showing it. Like for example, it's a very popular example, the egg color of Hawaiian bird. So if you cross the blue egg bearing bird with the white egg bird, Hawaiian bird, so you get F1 generation, which is heterozygous, right? And here blue color is dominant over white. So let's say you get 9 eggs, right? So if the 9 eggs, all the 9 eggs, they have the genotype that is heterozygous, right? But you can see they all are not showing the blue color. Even you can see some are colorless. Now, if you look at the colorless, you will assume that they have the genotype this one. Because colorless is only formed, white is only formed when you have the genotype recessive allele in homozygous condition, right? But this is not the case over here. So through these studies, we got to know, the scientists got to know that it is not only the gene which determines the phenotype, even environmental factors also play a very important role. There is a possibility some environmental factor not allowing this genotype to express fully, right? So we are not getting the blue color here, we are getting colorless only. So penetrance is that where an individual shows a particular phenotype which it is capable of showing. Then pleiotropy. So pleiotropy is where we have studied about the pleiotropic genes, where one gene influences several other traits, phenotypic traits. Like for example, you can see here G1 is the gene. It's a pleiotropic gene. So you can see it codes for phenotype P1. But again, you can see it is indirectly associated in generation and formation of P2 phenotype also. So if anything goes wrong with this G1 gene, like if G1 gene gets mutated, not only this phenotype P1 will be affected, but also the P2 phenotype generation will also be affected. So I have covered these topics in the class. Okay. So in the class, I've already covered these topics in detail. And then conditional mutation is where the mutation that has the wild type phenotype under certain environmental, permissive environmental condition, and mutant phenotype under restrictive conditions. And lastly, you have the phenocopy. So phenocopy, where the individual shows the feature which is characteristic of a genotype other than its own. And this kind of variation is produced not genetically but environmentally. So, like a very popular example, if you remember Venisa butterflies, we have discussed in the class, right? Even this cross veinless wing example I discussed in the class, right? And even the body color in Drosophila, right? We discussed this example in the class. So basically, in this particular question, it is mentioned that Drosophila larva is exposed to 37 degrees. Okay, the temperature. So adult flies that emerged had a cross veinless phenotype, and cross veinless is a known mutant. So basically, here we are talking about the wings, and we are saying like you can see this is the normal phenotype, the veined wings, okay, and this is cross veinless, which is a mutant phenotype. So the Drosophila, they show the normal phenotype, having this genotype, they show this phenotype, right? But there are other mutants of Drosophila which show this cross veinless phenotype, right? So we are representing it with recessive allele, that is CV, that you can see over here, right? Now, it is mentioned that when there is an adult Drosophila fly and you expose it to the 37-degree, it emerged that emerged had a cross veinless. So maybe this is having the normal phenotype, but because you provide a change in temperature, some environmental factor you provide, because of that, it results into formation of the cross veinless phenotype, right? So cross veinless genetically, it is for normal phenotype, but how it appears, cross veinless. And there is a mutant. In nature, we have a mutant of Drosophila which is naturally cross veinless, which is having this genotype. That is this one, right? So although this adult, because you exposed it to certain environmental factors, some kind of like, in the exam, they can give you some chemical reagent, some temperature, heat shock, right? So here it is 37 degree, the temperature, and it becomes cross veinless. But genetically, it was for the normal phenotype. So when this cross veinless is crossed with a mutant, so that means now this genotypically which is normal, but showing the cross veinless, when it is crossed with actual cross veinless, the mutant, the normal phenotype is observed. So normal phenotype means this one. So this indicates what? This is because of the concept that we studied, phenocopy. So although this Drosophila was having the genotype for the veined wings, but it resembles to the another organism which have a genotype for this cross veins. So this is about the phenocopy. So this is a very direct, the question based on very direct concept. So here the phenotype is acquired because of exposure to high temperature and not due to mutation in the gene. And therefore, such type of traits are called as acquired traits, and they will be lost when they are in the next generation with the change in condition. So here the answer is option 2. So those who have answered option 2, you are absolutely right. Phenocopy, Yusra, absolutely right. Okay. Now moving over to another question. Complementation. I'm sure by looking at the question, you can easily make out that this question is about complementation, right? So please go through the question and tell me what is the answer here. In Drosophila, recessive mutants A, B, C, D, E, F, and G all have the same phenotype, namely the absence of red pigment in the eyes. In pairwise combinations and complementation tests, the following results were produced. So this is the result you all can see, where plus represents complementation and this minus indicates no complementation. So which mutants have defects in the same gene? And 4 options are given to you. So tell me what is the answer over here. Now, such type of questions you get in part C. Very important. I know by seeing the question, most of you, like many of you, are not even trying to attempt it. But believe me, these questions are very, very easy. If you know the trick, if you know the concept, you can easily answer this question. So I want you all to try first. Okay. So what do you think? Where is the defect? So complementation, I showed you a part B question and I'm now showing you a part C question. So in part C, a figure will be given to you, right? And here you can see, we are seeing the genes and you have to determine by looking at this figure that which genes are complementing, which are not. Okay, Anupam, you are not able to understand the question. Fine, I'll explain it to you. And Sunshine and Surendra are saying option 1. So Anupam, just read the question once. It is mentioned that in Drosophila there are recessive mutants. We have A, B, C, D, E, F, and G. This is mentioned, right? They have the same phenotype. What is the phenotype we are talking about? Absence of red pigment in eyes. What phenotype we are talking about here? The eye color in Drosophila, right? By studying the concepts, I'm sure now you know that the red pigment is the wild type, right? The red eye color in Drosophila, it's the wild type. Absence of red color means it could be white, it could be apricot because these are the mutants, right? So absence of red pigment in the eyes, that is the mutants. So these are all recessive mutants, A, B, C, D, E, F, and G. Okay. Now, in pairwise combination, so these are basically alleles which are coding for this mutation, A to G, right? Now, in pairwise combinations in complementation test, the following results are produced, right? So you have to compare, okay? You have to compare A with B, A with C, okay, like that, or B with A, and like C with A, C with B, D with A, D with B, and so on. This is how you have to read the table. And one thing is mentioned that plus represents complementation, minus represents no complementation. So even just a few minutes back, we did a question on this concept only. That complementation will occur when? When the alleles of the different genes, when the mutation is in the alleles of different genes. Maybe there are two genes or more genes affecting the same character. Maybe let's say here, eye color in Drosophila, right? So when the mutation in the alleles of the same gene, they never complement. Mutation in the alleles of different genes only complement. I hope now you have understood the question, Anupam. Yes, Sunshine, you're right. Mutation in the alleles of same gene will not complement. Correct. So what is the answer here? Okay. Option 1. Rahul, Kantimati, you are saying option 1. Fine. Let's others also solve. And then I'll disclose the answer. Quickly. Okay, now you are able to solve option 1, Anupam. Okay. What about others? Ratna, what answer you are getting? Tulsi, option 1. Okay. What about you, Saujanya? What about you, Anu, Divya, Hina, Madhumita? Please let me know your answers also. Just try. In the class, we discuss the concept in detail. So, the mutation in the alleles of same gene will not complement, right? So, if you look here, The plus represents complementation and minus represents no complementation, right? So we'll start from here. So obviously A and A, so these are the same allele we are talking about. But if you see B and A, they are complementing, right? Then if you look at the C, it is also complementing with A and complementing with B also. Then if you see D, D is not complementing with A. D is not complementing with A, but it is complementing with B also, with C also. Then if you look at E, it is complementing with A, right? But and with B, but not with C, and complementing with D, right? Then if you, so this is how we read the table, right? That F is not complementing with A. F is not complementing with A, but complementing with B, C, and again not complementing with D, right? And G, if you look, it is complementing with A, but not with B, but complementing with C, D, E, and F, right? So what you can infer from here? We can infer that this A, D, and F are alleles of the same gene, right? Because they are not complementing, right? These three are not complementing. And even if you don't want to go read it this way, simply you can look for which alleles are not complementing. Like you can see D and F are not complementing with A, right? So that means A, F, and D must be the alleles of the same gene. Then you can look here that B and G are not complementing. If you look at this column, B and G are not complementing. So maybe these are the alleles of the same gene, but another gene. And then finally, you can see that C and E are not complementing, right? So that means this must be the third gene, right? Because now D and like D and F, it is already repeated over here. So no need of that writing it again. Then even so rest we know like F and F, it's the same allele. So the point is we are having three genes here. And out of these three genes, AFD is the allele of one gene, B and G are the alleles of the second gene, and C and E are the alleles of the third gene. So this means your answer is option 1. So yes, I can see majority have mentioned option 1. So I'm really glad to see the answers. That means now you understood the concept, right? And it's very easy. The question appears to be very difficult. It's just if you know how to read this table, how to read this figure, you can easily answer this question, right? So are you liking the concepts? Yes. Is this session really helpful? Please let me know in the chat. Are you really liking the sessions, the Brahma series? Is it really helpful for you? I'm not only talking about unit 8, but other units also. Please tell me in chat. Is it really helpful for you all? Okay, that's great. Thank you, Kirtana. Thank you, Rahul. Thank you, Sunshine, for responding. Glad to know about your views. Thank you, Sajanya, Surendra. So, yes, so those who don't know, I'm sure now you know about this WISE scholarship program. So, those who have joined just now and don't know about it, so this is information for you all that the Biotechnica has come up with this WISE scholarship. And the advantage of this scholarship is that under one scholarship, you get access to the class lectures. You be it CSIR net, be it GET, be it QED, be it IIT, be it GET, okay, and GATE also. And even you get access to the workshops, to the conferences, the internships, and international conference that going to be held in November. And also you will get access to the hands-on training. Right, we have a tie-up with several companies where if you enroll with us, you can get a chance to uh, go to these labs and perform, learn the techniques and perform in the lab also the techniques, the experiments on your own. So these are the different scholarships: 100%, 90%, 25, 80, and 50%. So for more details, you can call us on a toll-free number. Okay. So moving over to another question now. So here you can see another question. So it is based on complementation only. Given below is the result of a complementation test for 6 independent mutants, 1 to 6. Plus represents complementation and 0 represents non-complementation. So based on the above, which one of the following conclusions is correct? So mutations can be ordered in single cistron as 1, 3, 5, 2, 4, 6. So cistron is an alternative name for the gene only. Okay. Or all the mutations belong to single cistron, but their orders cannot be determined. But instead of single, now in this option it is mentioned there are 3 cistron. 3 genes are there. And maybe this 1, 3, and 6, these are the mutants of the, like maybe of one gene, 4 and 5 of another gene, and 2 represents the third cistron. And fourth is that there are 3 linkage groups. So mutations 1, 3, and 6 represent linkage, 4 and 5 represent another linkage group, and 6 represents another linkage group. So what you'll say, what is the correct answer over here according to you? I completely agree with you, Anupam. More you solve, more you learn, right? So yes, definitely, as I mentioned earlier, these type of questions are asked in the exam, and more you solve, so you gain the confidence, right? And even if such type of questions are asked in the exam, this coming exam, then in that case, definitely you can answer such questions and score as well. So 4 marks you can score. These questions are asked in part C. So quickly answer, because if you quickly answer, we are able to solve more questions. We can solve more questions, right? Okay. Option 3. So for option 3, I'm getting. Okay, all are saying option 3. You just have to compare. Compare 1 with 6, right? So here 0 represents non-complementation. So compare 1 and 6. Compare 1 and 3 because it is not showing complementation. 1 is not showing complementation with 6 and 3. Then if you look at 2, it is showing complementation. 3, not showing complementation with 6. 4, not showing complementation with 5. And, right? And then 5 is showing complementation with 6. So that means what? Maybe this 1, 6, and 3 must be the alleles of the same gene. 4 and 5 of another gene, and last we are left with only 2. So that means it is the third gene, right? And even I would like to elaborate the concept of complementation group over here. So those who don't know, like if you are confused between this concept, that what is the alleles of the same gene and what is the alleles of the different gene and how they are affecting the same character. So you can have a look at this diagram. See, as I told you here, one gene, one phenotype concept is not there. Here, many genes can be co-affecting the same character. This example I gave earlier also, eye color, eye pigment in Drosophila, right? So you can see this is the eye color mutations in Drosophila. Okay, so the genes are located on X chromosome. So you can see this carnation, garnet, vermilion. So you can see this is a chromosome and this is the location of the genes, right? So you can see carnation, garnet, vermilion. They are the mutant alleles of different gene. This is different gene. This is different gene. This is different gene. Ruby is different gene, and this white locus. This is different gene. And within this white, you can see we have buff, coral, apricot, and cherry, right? So these are the alleles of the single gene. So definitely they will never complement. If you're comparing apricot and cherry, two mutants coming together, they will never show wild type phenotype because they are alleles of the same gene. But if you are comparing apricot with vermilion, so yes, in that case, wild type can be there, complementation can be there because these are mutant alleles of the different gene. Okay, so a collection of mutations that do not complement each other, they form a complementation group. So this is even like here you can see that is what I'm talking about, that the apricot, right? So like you can compare like white with garnet, white with ruby, white with vermilion. So they will complement, right? Because they are alleles of different gene. But white with cherry, like here cherry, coral, apricot, buff, it will never show complementation because these are alleles of the same gene. Okay. So because here, as I mentioned in the previous slide, right, that these 1, 3, and 6 are not complementing, so they are present on one gene. They are alleles of one gene. 4 and 5, second gene, and second represents the third system. So here the answer is option 3. So I'm glad to see that all of you have answered it correctly. Now, quickly moving over to the question, which is based on the pathway. Even in the exam, you get these type of questions, right? Part C questions, that there is a hypothetical biochemical pathway for the formation of eye coloring insect. Two autosomal recessive mutants, A and B, are identified, which blocks the pathway. Now, please pay attention. Here, this A mutant is affecting this pathway, that is conversion of substrate X to brown pigment. And here, mutant B is affecting this particular pathway where substrate Y is converted to orange pigment, right? So intermediates are there, which are colorless in both the pathways. In one pathway, we have a substrate X, which is converted to brown pigment, and substrate Y, which is converted to orange pigment, right? And you can see that if both the pathways it will work, we will get a wild type phenotype, which is orange brown eye color. So it is mentioned that mutants are not linked. So considering that mutants are not linked, what will be the phenotype of the F2 progeny if crosses were made between parents of genotype? So they have mentioned the genotype of the parents, and the F1 progeny are intercrossed. So again, here to answer this question, you have to go step by step. You have to first find out that if these two parents, the genotype of these two parents which are given, if you cross them, what is the F1 progeny you get? And when you intercross the F1 progeny, what is the phenotype of the F2 progeny? That you have to find out. So just try solving it. I'm sure now you understood the question. So F1 generation and then F1 selfing of F1 generation gives you F2. So what is the ratio you will get? Will you get 9 is to 3 is to 3 is to 1, or you will get 9 is to 7, or you will get 1 is to 2, or 15 is to 1? That you have to find. Okay, Suresh, you are saying option 1 it is. Fine. Let's see what others answer is. If you understood the pathway, you can easily answer such question. Okay, Rahul, option 1 you are saying. Fine. Okay, Tilsi, you are saying option 1, Sunshine option 1. Okay, so uniform answer I'm getting. Yusra option 1. Okay, I really appreciate all the students who are really putting an effort and trying to solve the question. I'm really glad to see your responses, students. Very good. I don't need the correct answer. I always say that it is not important that you give the answer which is correct only. If you're able to find the correct answer, it's very good. But even if you're not able, if you're just trying, if you're not getting the correct answer, then also don't worry. Because see, unless and until you don't practice, unless and until you don't try solving the question, you will not understand you will not able to analyze your performance, right? Where you are lacking, right? And if you don't understand that, you cannot overcome your mistakes, right? The part where you are lacking, right? So it's good that you all are solving. Okay, Achal option 2, Anupam option 3, Kirtan option 1. Good, very good. Okay, Anu option 1. So we have very less time left now. Let's quickly let's get the answer. Okay, Vinushree option 2, Anupam option 1. Fine. See, students, I cannot give you more time because we have only 1 hour for solving the questions, right? So I told you if more like faster you respond, faster you give me the answer, more questions we can discuss in the class, right? In the live session, right? Okay, option 1. So majority says option 1, right? So let's have a check. So this is what the condition is given to us in the question, that this is the genotype of one parent and this is genotype of another. So we know that gamete formed will be by each parent will be this, right? And when you like when these two gametes fuse together, you get a double heterozygous condition and you get orange brown eye color progeny, right? Now when you perform selfing of F1 generation, so this is like a Mendel's dihybrid cross, right? But here, because a pathway is given, we have to see that whether are we getting the ratio that was expected from Mendelian dihybrid cross, right? Maybe sometimes there's a deviation from this ratio also. So here you can see that we have a substrate X which converted into an intermediate. So let's say there is a gene B which converts normal gene B, which converts the colorless substrate X into intermediate, which is again colorless. And then it is converted by the, let's say, normal A allele, which is converting it into the brown pigment. Now, we have the mutant here, right? So because of that, this pathway is blocked over here. And in substrate, like if you look at this pathway, the substrate Y is there and there is a mutant which is over here and it will block the pathway if we have this mutant allele, right? So basically for getting the orange brown, we will be needing both dominant A allele and B allele. So because of that, what will happen? This substrate X will give brown pigment and substrate Y will give orange pigment and we'll get orange brown eye color, right? So basically, when you perform the cross, these will be the genotypes formed by each parent, right? So I'm drawing a Punnett square. I've drawn a Punnett square here. And you can see here that if A and B alleles are coming together, right? Wherever the dominant A and B allele are coming together, there you have orange brown eye color. That is the wild type, right? So out of the 16 progeny, 9 you can see are orange brown. So wherever you have dominant A and B allele. So you can see I have mentioned orange brown, OB for orange brown. So wherever you have A dominant A and B allele, there you have the orange brown. So that is total 9. Now you can look here that here we have dominant A, but we have recessive B, right? We have the dominant A but recessive B. So basically we very well know that if we have the dominant A and we have the recessive B, so substrate Y cannot be converted to orange because this mutant, if it is present, it will block this pathway. But this pathway will continue because we don't have the recessive A here. Recessive A is not present in this genotype, right? So that means substrate X will be converted to brown, but the substrate Y will not be converted to orange. So because of this, we are getting the brown over here. So 3 are brown and you can see orange are those only where you have this recessive A. Because now what will happen because of this mutant allele, this substrate X will not be converted to brown, but substrate Y will be converted to orange. And therefore, the genotypes where you have the recessive allele A in homozygous condition, you get an orange phenotype. So one pathway is blocked, another is working. So whichever is working, that pigment will get. And lastly, this one you will get is the colors, right? Because in this case, both pathway X is also that is the substrate X is also not converted to brown and substrate Y will also not be converted to. And this is the reason answer here is option 1. So majority have answered it correctly. So very good. I really appreciate you all for answering the question. And even those who are not able to answer the question, I'm sure now you understood why here is answer 1. So this is all for now, students. I can see it is almost the time, right? So I'm sure you are liking these Brahma series, sessions of Brahma series. And again, tomorrow we'll be coming up with another session of Brahma series. The timing, you all know, it is 5 o'clock to 6 o'clock. So daily we have a session, one session, right? Each day we'll be having a session of of this Brahma series. So please attend these sessions. It is for your benefit only. And in any help you need, you can always contact us. So with this, I end my session here. So thank you everyone and take care.